Before starting the presentation, I would like to thank SECPHO for the opportunity to participate in this interesting webinar. So good afternoon, everyone. And my name is José Bengoechea, and I work as CTO at Nulab, and would like to give you a few details about the company. Nulab is a startup set up by CNTA, National Center of Food Safety and Technology in Navarra. Nulab wants to contribute to reduce food waste and food safety and quality optimization in the agri-food value chain. In order to achieve this goal, we develop and commercialize equipment and devices able to predict and monitor food safety and quality parameters on real time with accuracy and in a non-destructive way, digital environment throughout the entire production. Nulab has a highly trained team of more than 50 technicians to carry out many exploration studies and research the best technology in order to find the best suited device for product control. Definitely, Nulab will transform the sector revolution, revolutionizing the concept from sample to lab to sample. The companies in the food processing industry need integrated sensor systems to control the product at real time. This means that technology providers and digital solution providers have to work together to understand and grasp the needs of the agri-food companies and develop smart systems accordingly. From the other hand, accreditation and certification are also necessary to show compliance to food safety requirements. Finally, an integrated solution also means that the smart system transfers data from and to the production monitoring system. The current situation in the food industry is that the methods for quality control of the products are destructive, require previous preparation, qualified technicians, and appropriate facilities. We are working on the development of devices that allow the change towards online, rapid, reactionless, non-destructive, and non-real-time systems. The quality assurance methods used in the food industry have traditionally involved human visual inspection. However, like I said before, such methods are tedious, time-consuming, and sometimes inconsistent. In this presentation, I am going to focus on hyperspectral imaging technique. It has been regarded as a smart and promising analytical tool for analysis. We have taken advantage of benefits this technology offers when it combines vision methods based on red, green, blue color vision systems and spectroscopic techniques. I would like to explain quickly which properties we can obtain when we work with them. So, color vision systems have been successfully applied to evaluate the external characteristics of food. And spectroscopy is the science of acquiring and explaining the spectral characteristics of an object to describe light intensities emerging from its molecules at different wavelengths, and thus provide a precise fingerprint of that object. Optical properties of food products have been one of the most successful non-destructive techniques for quality assessment to provide several quality details simultaneously. Optical properties are based on reflectance, transmittance, absorbance, or scatter of polychromatic or monochromatic radiation in the ultraviolet, visible, and near-infrared regions of the electromagnetic spectrum, which can be measured by spectral instruments. Hyperspectral imaging allows for the visualization of different biochemical components based on their spectral signatures. So regions of similar spectral properties should have similar chemical composition. Well, this is a typical hyperspectral imaging system in lab. It has the following components: light source to provide illumination, usually provided by halogen tungsten lamps, detector which obtain both spectral and spatial information simultaneously, moving surface to hold and transport the sample, and finally computer to compose and store the three-dimensional hypercube. The data cube produced by hyperspectral imaging systems contains mass of information with large dimensionality. So the main purpose of hyperspectral data analysis are to reduce the dimensionality and retain the useful data for discrimination or measurement analysis of food quality and safety. That discipline, known as chemometrics, use mathematical and statistical methods. But this technology not only has advantages, it also has some drawbacks that we must take into account. The main analytical drawback is that it is an indirect method. That means that it needs standardized calibration and model transfer procedures. This modeling and data processing is time-consuming, and specialists are needed for calibration and standardization. During the development of a new technology applied to industry, we can distinguish three steps. Exploration consists of testing the technology for the intended purpose. We will carry out tests that show that the measurement of one or more parameters are accurate and comparable with similar results obtained by traditional methods. Second step is validation or demonstration of an existing digital solution in a food process technology environment. And finally, the application of this solution will be implemented as a tested digital solution in real environment inside the industry or production chain. Our knowledge and experience in agri-food sector allow us working from 2 points of view: food security, identifying critical points and microbiological control, and sensory and physicochemical quality. For example, control of quality specifications, defining quality standards, selection of quality product criteria. There are some applications in which we can use the hyperspectral technology, and we are involved ourselves in shelf-life product determination, control of sensorial features and physicochemical properties, and classify raw material at the entrance of the factory. We have currently assembled an offline hyperspectral prototype to measure freshness in a minimally processed vegetables plant. For this application, we will be able to classify the raw material and predict its useful life. Another success case is the determination of parameters related to the ripening of tomatoes and their organoleptic characteristics. In both cases, the correlation between sensory and physicochemical parameters has been essential. It's allowed us to identify those key instrumental analyses that give us sensory information. The great success of these applications has been the control of sensory characteristics throughout hyperspectral technology, being able to measure more than one parameter simultaneously with similar accuracy to traditional methods. Projects we are currently working on detect strange objects and detect pathogens. These applications have been included in a national project, as well as the determination of food fraud. It's a topic that we are beginning to tackle with this technology. It will be undertaken in an exploration phase within the framework of an European project focused on the detection of fish fraud. So that's all. Thank you for your attention, and if you have any questions, I will try to answer.